Okay, is it
All right,
working?
well, hello everyone. All right, Pierre,
Hello,
we're all
everyone.
yours.
Yeah, hello.
Okay, so we are going to answer all the questions that we couldn't answer to during the webinar. I would start by the beam parameters questions, and one was seems to be a very easy question to answer to, but maybe not that easy. Is about the dose limits. If it exists lower and higher dose limits, and maybe a difference between in vitro doses and in vivo doses. So what do you have to answer to that?
Yeah, well, in fact, if we are talking about lower dose per fraction and like doses around 2 grays, 3 grays, then the conventional irradiation is pretty good to protect the normal tissue. So there is no real added value to use flash irradiation. In that case, flash is providing the same type of tumor control and tumor growth delay, but there is no real added value to use flash. So it's really starting to enhance the differential effect in between normal tissue and tumors at pretty high dose, around 7 gray, which is a dose currently used for hyperfractionation regimen. So I would say that probably this lower dose is driven by the fact that we want to benefit at the normal tissue level, and this benefit is globally only seen around 7 gray. Now the upper limit. There is upper limits that is really dependent upon the organ and upon the volume of irradiation. So if you increase the volume, as for conventional radiation therapy, you're going to increase the toxicity. So at one point, you're going to reach a threshold. So it has to be explored. It has to be validated in any of the conditions that we want to use. So do you have something to add about that, Charlie?
No, I think that you're right. We need to evaluate the volume effects, as you said, for the in vivo studies, and it does look like the differences between flash and conventional in general require higher total doses to see differences in vitro than we do for in vivo.
Yeah, regarding the question of in vitro and in vivo, there is something that is really important. Before you go for any in vitro experiment, your beam needs to be validated in vivo. There is no other way to make it. You cannot validate a flash beam in vitro currently. We don't know enough to be able to do that. If you are irrigating normal cells and tumor cells, you're going to end up with the same type of pattern, flash or not. So here, what you need is an entire organism. Otherwise, you won't be able to validate the flash, your beam as a flash beam. Once it is done, go for it, Charlie.
Yeah, no, and I think too for the in vitro studies, it's pretty important to define the level of oxygenation your system's at, so that will help at least eliminate variability when different groups try to reproduce certain results like clonogenic kill or DNA damage, whatever it is you're measuring. So
right.
Beam parameters, volume effects, and in vitro, really pay attention to oxygenation, and then at least whatever data you get can be reproduced and replicated.
And to use a validated beam. Not to start with in vitro experiments. No. It's useless. Yeah, and then once your beam is validated, you just can do anything you want in vitro. Explore anything you want.
So this is one of the things that goes against what a lot of people have been trained in science in general, right? I mean, this is probably the only time in MCAT, and I have said this before, where you have an effect that's been defined in vivo. Instead of coming up from the basics and chemistry and physics, we're working our way backwards, and so it's a challenge for people to think in reverse like this. But you really have to define this effect in vivo, and then we can start piecing together the basic mechanisms. So it's a different way of approaching a problem. It's actually a lot of fun. So, okay, yes. Pierre?
I think the conclusion to that is that the flash effect is an in vivo effect and a biological effect. Yes. So we talked about in vitro experiments, and we had many questions about DNA. One was very interesting, and we didn't answer to that, about in silico experiments. So are there any reported FLASH biology studies with GN4 DNA or topaz?
Well, that's more modeling, and well,
Exactly.
yes, there is several groups that are working on that subject here in Europe and in the US. So right now there is a couple of publications that I'm aware of that are submitted, and some other projects that are ongoing, but I have not seen any published data so far.
No, I'm aware of some. I also have a few manuscripts I'm looking at, but I really can't comment on that publicly at the moment.
Yeah.
I know there's groups working on it. So I mean, if the question is there anything published that's really illuminating regarding the modeling on this? And not just yet, but it's coming.
Yeah, yeah, it's going to be coming. The important point is to be able to parallel those experiments with real data, real measurements.
Okay. Now we have more biological and technical questions. One question about the normal tissue studies is about the learning and memory, and if there are sex differences during learning and memory tests in mice.
Well, the answer to that is without question, yes. The sex differences are very dependent on total dose, and what particular clinical or experimental dosing paradigm you use. For most of the studies we do in our lab, we group them together and use suitable ends so that really a lot of the differences are averaged out. But to assume a priori that the neurocognitive response of males and females is the same is inaccurate. And so there's a lot of interesting work to be done with FLASH and sex differences. At present, there's no conclusive data indicating that FLASH will be markedly different between males and females. But that is something that's a valid question and needs to be investigated further.
Yeah, and then we're back to the definition of upper limit threshold.
Yeah.
That could be slightly different in between the females and males.
Mm-hmm. Radiation sensitivity, etc. About radiation sensitivity, what was the age of at the time of irradiation for juvenile studies?
They were radiated 3 weeks of age.
Okay, and another question about this this study. The attendee was curious to see if testing juvenile mice when they reach maturity to see the long-term effects of flash irradiation. Have you done that?
So that's a good question. So the animals were irradiated young. In some of the data that we presented yesterday, keep in mind that the animals were actually tested at the age of 2 and 4 months, and the neurocognitive deficits persisted at least to the point of 4 months. There's always a question that I get: how persistent are the radiation-induced decrements? And there's, of course, you can always keep the animals longer and test them. There's really nothing that we've come across in the literature or in our experience with either juvenile or adult mice that suggests that these effects ever resolve. And so at least over the course of testing and experimental paradigms published in preclinical models, the radiation-induced deficits are for the most part permanent. And left on their own device, they really don't get better. And this fits with what we see in the clinic too. People don't all of a sudden get better after the radiotherapies. They're stuck with these problems, and it really is one of the major unmet medical needs in the field. And so whether or not these animals would recover, 8, 9 months, a year later, that remains to be seen. But at least over the course of our experimentation, animals that are given conventional radiotherapy remain impaired over the course of our experiments.
Yes, indeed. Another question about learning and memory. The brain function discrimination indices in the results you show seem to have a lot of variation between experiments. How do you explain that and control for these variations?
Yeah, that's really true. Well, I didn't let Charlie answer in a more definitive way.
Yeah, so for those of us who are engaged or invested a lot in neurocognitive testing, it's very different kind of experimentation than running a Western or doing looking for DNA repair foci. There is always going to be inherent variation in how mice explore and interact with their environment. So the fact that there's variation in neurocognitive testing is the nature of the beast, so to speak. You must control for it. And one of the things that we always do with every experiment is we always run concurrent controls. So if we run an experiment in January and we want to look 6 months later and we want to replicate that maybe later in the year, we always have to include the unirradiated groups because that really forms the basis of comparison. And they're taken through the exact same experimental paradigms, habituation, and test phases. So if anything, so to speak, weird happens, then all the cohorts are exposed to these same conditions. And there is a natural variability, no doubt. And you can control for that. You can track locomotion and all this. And we video record everything. And every now and then, the controls don't like to do the test. And this is an inherent confounder in behavioral testing.
Yeah. In addition, in the experiment, I showed the animals were bearing a tumor, which is a very huge confounding factor.
Well,
and in fact, we all know that from one experiment to another, the tumor growth is not completely the same always. If we do have something that has happened in the animal facility, some kind of noise or whatever, the variation for the recording can be important. The only way to go over that is to increase the number of animals.
Right, and remember too, when you do the neurocognitive testing in the presence of a tumor, really our groups have been the first, really the only groups to successfully carry this out, that you cannot expect these animals to carry on for 6 months because they're going to either die or the tumor itself will cause the problems. And so I guess my advice would be, if you're going to do neurobehavioral testing, tumor-free or tumor-bearing animals, you need to run concurrent controls. You're going to set yourself up for failure if you rely on historical controls. So always have a control cohort, and then at least you set yourself up to get the best data that you can.
I think this was a very clear answer. Then we have questions about the tumor studies. So what was the dioxygen concentration in the tumor for the hypoxia and hyperoxia experiments?
That's
used, and how actually the hypoxia and hyperoxia were checked?
Yeah. That's an interesting question. In fact, we try to do direct oxygen measurement with a probe that is called oxalite. And in fact, when we happen to work with this probe in the subcutaneous graft, we were never able to measure any type of oxygen concentration. And we think that it's related to the fact that those type of tumors are very dense, whereas the probe, by the way, works very nicely in any other normal tissue or in any other tumors that is not subcutaneous. And we think there is a problem, a pressure problem. We're not 100% sure about that. So we have not been able to measure directly the oxygen concentration inside the tumor. What we have been able to measure on one hand is the oxygen supply with the carbogen supply or the clamping, and we've been able to measure the hypoxia using some hypoxic staining and an endogenous marker that is called pymonidazole that enable us to show that indeed in the clamp tumor, all the tumor is completely positive for pymo. In normoxic, we do have around, you're going to perhaps correct me, Pierre, because you did those experiments, but around 60% of hypoxia, and in carbogen, everything is completely non-hypoxic, not pose. So it was the way we did it.
It was really clear on the results, yes. What kind of glioblastoma cell lines did you use during those experiments?
I think you can answer this one better than I
will
can.
answer this one.
Yeah.
So we've studied different glioblastoma cell lines. For all subcutaneous tumors, we've used U87MG cell line, so human glioblastoma. And for the brain tumors, so the autotopic model, we used a model that was developed by Doug Hanan in Lausanne, which is called H454, that comes from genetically modified mouse model that develops spontaneous tumors. A cell line was derived, and that's the one we used transplanted in immunocompromised mice. We also have glioblastoma models that come from immunocompetent mice, and we are doing experiments currently with those models. So we have kind of broad panel of glioblastoma cell lines that we can use for these experiments. I don't know if you want to add something.
No, very good.
Okay. We have an interesting question about the fractionation studies that we've done. Given the indication of effects of fractionation, it would be interesting to look at conventional fractionation regimen like 30 times 2 gray. Is it possible to do that in mice? Is it interesting to do that in mice?
I have. Well, I'll let Dr. Vosanone answer, and then I'm almost certain if she doesn't say something, I will too about that. But you want to? Well.
No, I think I just explained in a very clear-cut way what we did and what we could do, because we had some animals that were implanted topically in the brain with tumors, and then each time we do irritate them, we need to anesthetize them. So there is a balance in between what we can do and what we cannot. And if we kill the animal after the fifth fraction, it's useless. So that's why we went the closest as possible to the clinical fractionation. That was 3.5 gray, and we could anesthetize the animals only for time over a period of an interval of 24 hours, so 4 days in a row of anesthesia in an animal bearing a tumor. It's already pretty good. Plus, as I said before, going to retrofraction is just of no real use, because we know that conventional irradiation is sparing to normal tissue in that situation. So we don't have any additional benefits.
No,
we're gonna end up with the same type of curve.
And for those of us that do animal work, you're not going to anesthetize an animal 30 times. There's just no way.
No, the only thing you can do is you can do. Eventually, you can do it sub-Q. That's an option.
Well, and after that level of anesthesia, I can tell you that you're going to induce neurocognitive impairments. And so you're going to have a very hard time with your control group.
Yeah.
So it's just, so this goes, we cannot do exactly on animals what we do in people. And so that we do the best we can, but we can't do a 30 fraction regime of 2 gray with animals.
Well, we could make it sub-Q.
Yeah.
More than a question of feasibility, is it interesting to do it 30 times 2 gray? In the context of glioblastoma, for example, when we know that conventional irradiation is harmful for the brain, but on the other side, this 30 times 2 gray is not really efficient on glioblastoma. I mean, the patients still die after 14 months post-diagnosis. Is it interesting with flash irradiation to study that, or do we prefer to go somewhere?
Was the question only focused on the glio? It's not.
Well, 30 times 2 gray, I guess, is for the glio, but
yeah.
For the glio, no, it wouldn't be of any interest to me at all. I mean, so look, we haven't been very good at helping these people, and doing things the way we do is simply not working. And so I think the trend to making these people feel better in the context of FLASH is certainly moving to a hypofractionated regime, increasing the dose of the tumor bed while we minimize normal tissue effects. That's where we want to make a difference. And so,
but on the other hand, I can see the experiment being done sub-Q with 30 fraction of 2 gray to be sure that we're not protecting the tumors there.
It can be interesting.
well,
That's
Indeed.
something we could schedule.
yeah,
Indeed.
orthotopic tumor is not going to work for that.
No.
Brain.
No, for sure. No. We did the best we could for the orthotopic,
Yeah.
but sub-Q we could, and at least. But then I guess if we want to show the ISO efficacy, we're gonna need a crazy amount of mouse statistically.
Think about that experiment. You've got 30 days in a row of irradiation and anesthesia.
No. Then we can do without anesthesia in contention. Oh no, with anesthesia, it's no way.
Yeah.
No way. But if we are doing sub-Q, we can put the animal in a restrainer, and
Yeah,
then
sure.
That's feasible. That's feasible. But yeah, if we look at the power calculation to do that, I guess we're gonna have to include over 100 animals in each arm.
So I
Yes, the
think what you're
questions- the
saying is
questions
you
remain
need to get a grant funded to do this, Dr. Vosino.
Yeah.
Very good.
And we have a last question about fractionation. Are the fractions given daily? And I will add also, is it important to give the fraction daily, or have you studied other schedules?
Yes, that's what I said. Usually we give the fractionation about the different fraction 24 hours apart. But in the 3 times 10 gray experiments, and I think I mentioned that, the interval was 48 hours. So yes, the interval is also something we need to study. So some really systematic type of investigation is going to be needed, that's for sure. In addition to everything else. Yes.
Everything else. I think there are many questions that remain open.
I think for the people listening and or interested, there's a lot we have to do still. So we reach out to our colleagues to do some experiments and get some good data sets so that we can all move forward and decide what the most important parameters are at the physics, chemistry, and biology level. So, and the quicker we have more groups that are validating or not this flash effect, the quicker we can try to get this in the clinic and do some real good. So
yeah.
Okay. Well,
all right. Well, listen,
I think
thank
we
you,
answered
Pierre
all the
and
questions.
Dr. Vosino.
Excellent. Good.
All right.
Thanks to both of you for your interesting answers.
All right.
Thank you. Thank you.
Take care,
Ciao,
everybody.
ciao.
Bye.
Goodbye.
Take care.